everyone, today I'm going to show you how to do cell passaging. So cell passaging is to prevent cell from overgrowth. So when cell is reaching like 100% confluency, we need to separate them to more dishes. The more cells in log phase, the more viability you can get. So let's start it. So before the experiment, we need to pre-warm the reagents using 37-degree water bath. So the reagents we need today is the complete medium and the sterile PBS, and also the dissociation medium reagent trypsin. So the first step is to remove the old medium. So I'm going to use a glass pipette and connect to this vacuum system and get rid of the old medium. So after getting rid of the old medium, I'm going to use 2 milliliters PBS to wash the cells. Because the cells is attached to the bottom of the dish, so make sure you add the PBS slowly against the side of the dish. This will prevent interruption of the cells attached to the bottom. So just gently swirl the plate to spread the PBS. And then remove it. So the next step is to use the dissociation reagent, the trypsin, to detach the cells from the bottom of the dish. Also gently swirl the plate to spread the trypsin. And then remove the majority of trypsin. And use the leftover to detach the cells from the bottom. So I'm going to put the dish back into incubator for 1 minute incubation. So after 1 minute, you can take out the dish from the incubator. And we can also gently tap the dish to disassociate the cells from the dish bottom. So can you actually see the cells are sliding off from the bottom of the dish? And we can also visualize the cell's morphology after digestion, after detachment from the bottom. So we can see the cells right now are round shape instead of the spreading shape before the trypsin digestion. So after digestion, we need to add complete medium to stop the digestion. We can further pipette the cell solution up and down to make the cells form single suspension. Then we transfer the single cell suspension to a centrifuge tube for centrifugation. See the cell pellet at the bottom of the tube. After centrifugation, we can remove the medium from the tube. This step is to get rid of the trypsin reagents and the leftover of the old medium. And then we resuspend the cells using fresh medium. And we use the serological pipette to gently pipette up and down to resuspend the cell pellet. And we transfer the cell solution to more fresh medium. for passaging. So I'm now spreading the cell solution to more smaller dishes.